What is gender? I don't know. They keep changing the definition. At one point in time, the term gender was used interchangeably with sex, male or female, masculine or feminine. It was sometimes used instead of the term sex to avoid implying the sex act. In 1970, things began to change. Some academics began to define gender as a social construct. That is, gender is defined by the characteristics that a given culture perceives as masculine or feminine or some other gender. As the combination of characteristics grew, the number of defined genders increased. Some sociologists have defined as many as 72 genders. And some are now saying that a person's gender is whatever they say it is. It depends on how they feel about themselves and their sexuality. If a gender is determined by feelings, then it's a property of the brain. Some say that the gender of their brain can be different than the gender of their biological sex. It's getting pretty complicated, so I'm not even going to try to attempt to define gender for you. Instead, I'll describe many things that can influence the development of sex organs and the brain from the time an egg is fertilized until it is an adult human being. Then you can use this information to help develop your own definition of gender. Good luck! In all mammals, including Homo sapiens, there are two and only two types of gametes. There are eggs, which are large and contain not only a nucleus with chromosomes, but also cytoplasm, with all of the organelles and dissolved materials that are necessary to keep a cell alive and well. The other type of gamete is the sperm. Sperm are very small, not much more than a nucleus with chromosomes and a mobile tail. By definition, the body type that produces eggs is female, and the body type that produces sperm is male. Each gamete contains one complete set of 23 chromosomes, one of which is a sex chromosome. The two types of sex chromosomes are called X and Y. All eggs contain an X chromosome. Sperm can contain an X or a Y chromosome. When a sperm fertilizes an egg, the resulting cell is called a zygote. Zygotes with two X chromosomes develop into a female. Zygotes with an X and a Y chromosome develop into a male. At least that's how it works most of the time. But oh, so many things can go wrong. And even if nothing goes wrong, every individual zygote is unique, and each develops in its own unique way. We will discuss many of the events that can influence the developing individual. You will be hearing the terms DNA, chromosome, and gene, so it is best to clarify them before we use them. DNA is a very long helical molecule that contains genetic information in the form of a sequence of 4 different compounds called bases, represented as crossbars in the helix. The DNA strand wraps around special proteins. Before a cell divides, the complex coils once more, condensing the DNA. This coil then folds back and forth many times and eventually forms the chromosome. A gene is a section of the DNA. The beginning and the end of a gene are not visible. Fortunately, however, specific proteins in the cell recognize the beginning and end of the genes and control their activity. Sometimes the chromosomes carried by the egg or sperm bring with them errors that have occurred in the past. Errors can come in the form of mutations, which are defined as changes in the sequence of the bases in the DNA. there could be mutations in one or more of the genes that influence sexual or brain development. We will look at two examples of mutations that affect the development of sex organs in a fetus. Then we will look at examples of non-disjunction in which the X and the Y chromosomes are not distributed properly during gamete formation. The SRY gene, located on the Y chromosome, codes for a protein that controls several genes involving the development of testicles and other male genitalia from embryonic tissue. In the absence of the SRY protein, the gonadal tissue will develop into female genitalia. Thus, because XX embryos have no Y chromosome, the gonadal tissue always develops into female genitalia. A mutation in the SRY gene can modify or prevent the production of the protein. The gonadal tissue in the XY embryo will then develop into the uterus and fallopian tubes, but the ovaries will not be functional. Hence, a child will appear to be a female, but when approaching adolescence will not produce estrogen or go through puberty. The androgen receptor gene, located on the X chromosome, codes for a protein called androgen receptor. Androgens are a class of hormones, including testosterone, that are necessary for male sexual development. They are active during fetal development and puberty. Each androgen hormone must bind to a receptor in order to function. Many different mutations in the AR gene have been detected. For example, mutations can reduce or eliminate the function of the receptor proteins. Thus, in an XY embryo, no matter how much testosterone is present, without the receptor, male sex organs will not develop. The external genitalia will appear to be female, but internal female sex organs do not develop. During gamete formation, X or Y chromosomes are sometimes distributed incorrectly. The resulting zygotes are usually viable and develop into adults. The most common resulting syndromes are Turner syndrome, XXX syndrome, Klinefelter's syndrome, and Jacobs syndrome. Females with XXX syndrome are usually fertile, but individuals with any of the other three syndromes are nearly always infertile. Individuals with any of these syndromes can appear normal and function normally. However, when data for several traits are determined, the averages are often different than the averages for the general population. For example, females with Turner syndrome are on the average short, have a webbed neck, have elbow and wrist deformations, and sometimes have cognitive deficits. Females with XXX syndrome often have developmental, psychological, or behavioral problems. Males with Klinefelter syndrome often have a higher risk of male breast cancer, have a lower IQ, are often immature, shy, insecure, anxious, and have poor judgment. Males with Jacobs syndrome are often very tall with large heads and low muscle tone. They sometimes have developmental delays and behavioral issues, and they are prone to having asthma and are often autistic. Mutations are not all bad. Most mutations are neutral. Often, a mutation or a change in the DNA does not cause any observable change in the individual. Even if a change does occur, it might not have any effect on the individual's ability to survive and procreate. For example, an individual's eye color has little or no effect on a person's daily living. Some mutations are good. For example, infants have an enzyme called lactase, which breaks down lactose, the sugar found in milk. In most mammals, the production of lactase decreases after infants are weaned. But at some point in time, a mutation occurred that maintains the production of lactase into adulthood. Our hunter-gatherer ancestors had no source of milk after they were weaned, so the mutation would be considered neutral. However, the mutation was advantageous for pastoral cultures. And today, with milk available in supermarkets, the mutation is advantageous in our modern societies. Nevertheless, fewer than half of the population received the mutation and therefore are called lactose intolerant. All genes carry mutations. Over hundreds of millions of years, millions of mutations created the tremendous genetic variability that we see in living things today. Humans have approximately 25,000 genes, and every gene comes in more than one form. Different forms of the same gene are called alleles. For example, the gene for eye color has an allele for pigmented, which results in shades from green to brown, and an allele for non-pigmented, which results in blue eyes. Some genes have only 2 alleles, while other genes have a dozen or more alleles. Every person has 2 alleles for each gene, one from each parent. Of the 8 billion or more people on Earth, no 2 individuals, other than identical siblings, have the same combination of alleles for all genes. Hundreds of genes influence the development and maintenance of sex characteristics, both the physical organs and behavior. Some genes influence the activity of other genes. Hormones influence the activity of many genes. Genes function in an environment created by the product of many other genes in the cell, and this environment is unique for every human being. Try to visualize that in a cell, genes involved in sex characteristics, as illustrated in blue, are functioning in a sea of all other genes and gene products. Genes carry the information that guides the development of a zygote through the embryonic and fetal stages and throughout the life of the individual. All embryos are the same until week 6 or 7, at which time, in the XY embryos, the SRY gene starts the process of the development of testicles. At approximately 8 weeks, the fetal testicles start producing testosterone. The maternal ovaries and placenta produce estrogen. The sex hormones stimulate the development of the reproductive organs in the fetus. Estrogen and testosterone also influence the development of many parts of the fetal brain with respect to relative sizes and functions. But before we start comparing sizes of anything, it's helpful to review the concept of the normal curve. When plotting measurement data, you usually get a normal curve. When comparing data from 2 populations, you're really comparing the averages of those 2 normal curves. The heights of men and women provide a good example. When you say that men are taller than women, you're saying the average height of men is taller than the average height of women. However, some women are taller than the average height man, and some men are shorter than the average height woman. Also, the distribution of some curves is wider than that of others. Now back to the influence of the sex hormones on parts of the brain. Here are a few of the comparisons that have been made while studying the brain. The hippocampus is larger in women, while the amygdala is larger in men. Women have a slightly larger frontal and paralimbic cortex, while men have a larger parietal lobe. Women have a greater density of nerve cells in some language areas of the brain. The normal curve also applies to brain functions. For example, women are more likely to have anxiety and depression, while men have a greater risk of schizophrenia. Men tend to be more assertive than women and to have a higher self-esteem. Women are typically more extroverted, trusting, and nurturing. Men would rather work with things, while women prefer working with people. Men are better at visualizing spatial skills, while women have better verbal skills. The environment in the womb affects the development of a fetus. For example, very poor nutrition for a fetus can result in a low birth weight and physical problems such as cardiovascular disease or insulin resistance later in life. Excessive stress on the mother can affect the brain development of the fetus, resulting in emotional and behavioral changes later in the life of a child. Also, the female fetus of mixed-sex twins is exposed to testosterone from the male fetus. Some evidence indicates that this exposure can masculinize the female twin anatomically and behaviorally. Early infant development. The most rapid and critical period of brain development is between birth and age 3. The experiences and interactions with adults during this time have a major and lasting influence on brain development. Developing attachment with a caregiver in the first few months of life is critical for an infant's sense of safety and protection. Lack of proper attachment can cause difficulty in forming relationships throughout life. The sound of cheerful voices, gentle touching, and good nutrition help prepare an infant for a mentally and physically healthy life. Unpleasant experiences during these formative years make it more difficult for the child to cope with crises later in life. Puberty is a very turbulent time in the adolescent's life. Sweeping changes take place in the body and in the brain. A hormone secreted by the hypothalamus in the brain stimulates the gonads, that is, the ovaries or testes, to release large amounts of sex hormones, estrogen or testosterone. These sex hormones trigger the development of secondary sex characteristics, such as breast development in girls and facial hair and voice deepening in boys. Both sexes experience growth spurts. The rate of brain development increases sharply. Estrogen and testosterone influence the rearrangement of neuron circuits in the brain. Some of the changes affect the body's response to these same hormones throughout adulthood. If these changes fail to occur during puberty, it could cause problems for the remainder of the person's life. Some of the changes in the brain are important for learning, memory, and the processing of emotions. These hormones stimulate sex drives. The rate of brain development peaks around 13 to 15 years of age, but continues to about age 25. The last part of the brain to fully develop is the prefrontal cortex, the area that executes decision-making, judgment, and problem-solving. The atmosphere of the home in which a child is raised impacts the child's development and attitudes about themselves and about life. Was the father angry and abusive, or was he caring and compassionate? Was he even present in the home? How might this affect a child's attitude towards men in general? Was the mother nurturing and attentive, or was she negligent and distant? How would this affect a child's attitude towards women in general? Were the parents fervent believers in a fundamentalist religion that preached purity culture? Being taught that sex outside of marriage is sinful and evil and will send you to hell will often distort a child's attitude towards sex in general. Who were the adolescent's peers when their brain development was at its peak? Peers have a great influence on a teenager's self-image. Back to gender. Do you have a better idea of how you would define gender, or are you more confused? Is the term gender best defined as interchangeable with sex and used to avoid confusion with the sex act? Is gender a social construct determined by society? Is a person's gender whatever they say it is, as determined by their feelings? You have seen how genes determine the type of sex organs and genitals that develop in a growing fetus. You have seen the myriad things that affect the development of the brain, and that the brain is not fully developed until a person is about 25 years of age. If gender is a social construct, how does society define gender during all these stages of development? You saw the tremendous and rapid changes that take place in the body and the brain during puberty. If feelings determine gender, can a person's gender change during all of these stages of development? More than 8 billion people are living on Earth today. No two are genetically the same except for identical twins or triplets. No two people have had the same experiences from the womb to adulthood. Is there a best way to categorize 8 billion unique individuals into a small number of specific genders? Will you define gender according to biology, sociology, or psychology? Are there 2 genders, or 72 genders, or possibly 8 billion genders? What is gender? What is your conclusion?